Good afternoon. We're going to go ahead and get started. Welcome back to Track 3, Drug Development Pathway, sponsored by PRA Health Sciences. Thank you to Pfizer for sponsoring this afternoon's lunch. I am Cassandra, and I will be your Track 3 room host today. As a reminder, I would like to welcome those joining us remotely via the live stream, and remind all of you to download our mobile app, which we will be utilizing at the end of this session for the Q&A. For this session, we are joined by Tim Miller, CEO and President of Abeona Therapeutics, and Ellen Morgan, CEO of Agility Clinical, who will be talking about understanding the constraints on clinical trials, inclusion criteria, control groups, and blinding, and more. Please help me in welcoming our speakers.
All right, so I think I'm going first.
I think so.
All right. Make sure everybody have coffee. I think we did this yesterday. I see a couple thumbs up over there. All right. Well, welcome to Understanding the Constraints on some clinical trials. So Ellen and I are going to go through a couple of slides to kind of help align everybody on what clinical trials are, some of the things about what goes into a clinical trial, and then just some of the other issues about what you look at from an academic perspective, from an industry perspective. And again, just we're happy to start taking questions as well. So I'll start us off with clinical trials go back a long way ago. All right, so it's my own slide, and I'm going to have a hard time to read it. So I'm going to walk over here. If you guys will, this is one of the great things about having a floor mic, because I can walk over here and talk about it this way. So again, from the Persian physician and philosopher, going back to over 1,000 years ago and more. The clinical trials are something that was known that you wanted to try an experiment in medicine to make patients better, right? And bring up the famous clinical trial from about citrus fruits and scurvy, because if you get scurvy, your teeth fall out. So somewhere along the line, they found out that if you had vitamin C on board, that you would be able to recover from scurvy, and so that's one of the reasons why people brought oranges and limes and lemons and stuff onto ships. So, but these rules for clinical trials, go back a long way. And for the experiment and testing of drugs, they said that drugs must be free of any extraneous substance. So there's going to be some level of purity that goes into the drugs. There's got to be used on a composite, or not just simple, okay? But it must be used on a simple, not a composite disease. And the drugs must be tested in at least 2 different types. So again, we start talking about the quality of the drug and the strength for the disease. We talk about the time of action, the effect of the drug, and most importantly, that experimentation must be done with the human body. For testing a drug on a lion, I have no idea if anybody tests lions anymore, okay? Or a horse may not prove anything about its effect on man. So again, even many, many years ago, this was set down, some of the things about clinical trials designed. And then, you jump forward, a lot of years, and we start talking about the father of immunology and smallpox, and Edward Jenner, and how he started to treat drugs. And this is actually a really classic story. I won't go into all the details, but he basically injected a pediatric population, a boy, and again, the boy ended up surviving. But again, very early clinical trials. So we jump forward now and talk about why some of us are here today, and talk about rare disease trials. And rare disease gene therapy trials right now account for less than 5%, okay, of the clinical trials that are out there. And you can see cancer, takes up the lion's share of clinical trials. But rare diseases, they're the zebras. We like to talk about things that are unique. I wore my special zebra socks today, okay, showing that we're all zebras in one form or another. But rare disease trials, why are they different? Well, we have small populations, and we have limited opportunity to study right there, and we have some very small populations to study both safety and efficacy. We're looking at, generally speaking, wildly or highly heterogeneous populations from these treatment centers. And within these diseases, you have some natural variability, and then outside of these, when you start talking about a class, lysosomal storage diseases, that's also very, very broad. So there's my zebra right there. And these diseases are often poorly understood, often because of the mortality. When we talk about some of the MPS III, for example, 70% of those kids don't reach the age of 18. So, as a small anecdotal story we talk about sometimes is that, many of the people in the rare disease space, we knew that this was, MPS III was a rare disease. It was a pediatric disease. And when we ended up trying to get the orphan product designation with the FDA, they found one paper that talked about an attenuated form, and those kids were all older than age 18. So they said, it's not a pediatric disease. So we worked with a number of our foundations to go in and help us put together a registry, and they ended up coming back and saying, okay, we believe you now. And it was, again, a great example of us working with foundations and working with the FDA as well. So, these diseases, while they're poorly understood, there's often not a lot of natural history study data, which is why you do natural history studies as part of the clinical trials process. So when working with the FDA and working with a lot of the foundations, talking about, hey, natural history studies, we need to do this, okay, to be able to get an idea about outcome measures. And we'll talk about that in a little bit. Many of these are life-threatening diseases. They have unmet medical needs. And again, I'm not telling anyone in this room, that is a surprise. But there are also, many of them affect pediatric patients, and those put us into an additional constraints on, how we deal with treating in a clinical trial, the ethical reasons for, treating kids. So for those of you that don't know about how you go from bench to bedside or concept to clinic, you hear a lot of these catchphrases and things like that. We start talking about what's this path? Well, when you move intellectual property from concept to a clinic, you go through, generally speaking, what's called an IND. Okay, and the next slide talks about some of the jargon that we use in clinical trials. But that's an investigational new drug application. So you start very, very small in vitro, so in a test tube. You move up to small animal efficacy, then you move into potentially small animal safety. Then you start to get through the FDA with a pre-IND meeting, and you do some large animal safety studies. Then you go into an IND for submission, and once that gets allowed, things don't get approved at this stage. If they get allowed, you can start your clinical trial. But really what you're trying to do is, get the proof of mechanism that demonstrates that the drug interacts with, the intended target to have a level of efficacy. So here's some of those acronyms that, Ellen and I are going to kind of talk about a little bit back and forth today. We'll talk about IP or the FDA, IRB and IBCs. The IRB is usually a body that's governed at the hospital. They're responsible for all of the hospital patients' safety and really some of the ethical reasons about how and why you go into a clinical trial. We talk about CBER, okay, which is the Center for Biologics Evaluation and Research. Many of the drugs that are being developed by the companies, that are presenting out there, they go through CBER. And then CMC, that's a thing that people generally use as chemistry, manufacturing, and controls. That's when you talk about the drug manufacturing portion of your clinical trial. And GMP, you hear that thrown around a lot too. GMP is good manufacturing practice. There's a whole set of rules and regulations that talk about, your drug needs to meet these criteria, okay, to be able to go into patients. So really there are two types of clinical trials in general. There's the observational type and there's the interventional type. So observational, generally these are natural history studies, and interventional is where you're trying to go through some level of treatment. Natural history studies, again, evaluate how these disease progresses. Again, no surprise here. But there's a number of other types of models out there, from prevention, screening trials, diagnostic trials, even, things where you want to get to eventually is a treatment trial. And at the end, there's also these quality of life trials that are supportive care that try to find ways to improve patient comfort or quality of life. So again, we talked about the phases of the trial, right? We talked about how you got there, from talking about all of your in vitro work to up through your pre-IND meeting with the FDA to going through your safety studies and then onto your IND. Once you get through your IND, then go into the phases of clinical development, from phase 1 to phase 2 to phase 3 to potentially market entry, and then phase 4 post-marketing approval studies. So again, these are small, and again, as you move upwards, traditionally you need a lot more patients going into these. Now, rare disease is different, and we generally talk about the accelerated approval path. And when we're talking about 15 or 25 patients, that you might be able to use and have data from, to go to the FDA or the EMA for a marketing approval, it's a lot less patients, possibly because these are rare diseases, but that puts additional constraints on what you need to show and how you need to show it. These aren't often going to be 500 patient trials. And then we talk about some of the constraints around the clinical trial design. So give you an example from Abiona's programs. We are looking at MPS IIIA and IIIB, but our minimum age is 2 years old. okay, for a reason that, when some of the outcome measures that we learned in natural history studies allow us to enroll patients that are 2 years old and older, because that's where the neurological exams are. They're not necessarily valid or applicable to kids that are under those 2 years age. So that's a constraint that you have to deal with in some of your clinical trial design. And we talk about randomized. These are schemes that you use to assign one patient group to the other, whether they're going to get the low dose or the high dose or the mid dose. Non-randomized means you're just straightforward going through on a straight enrollment. Blinded, you hear blinded or double-blinded a lot. Okay, and that has to do with whether a patient knows that they're getting a drug, or whether the patient and or the study staff at a hospital know that they're getting a study drug. And of course, placebo means that there's inactive with no therapeutic substance. So we look for in these early phase clinical trials, proof of concept. And again, we'll use a kind of case study as some of the Abeona work right now, where we've gone through 6 patients so far. We've demonstrated some success that we call that early efficacy, or signs of early efficacy. But we're trying to move those forward now into these pivotal trials, where we're enrolling another 8 to 10 patients, and it then becomes more efficacy oriented and less safety. Usually you see dose escalations around that type as well. You're trying to focus on critical questions, such as that you're trying to drive your decision making with data. Do you go up a dose? Do you change your route of administration? How do you rapidly try to get from a fast to fail to a fast to market? Because the rare disease has so little out there for approvals, for treatment, these have a different, somewhat standard which they're held to. You do see faster development for global registration. And you're trying to focus on things that are going to give you a higher probability of success. So you're trying to choose the right vector for the right disease or the right injection method. INDs, as we call them, investigational new drug application, these are governed by the FDA 21 CFR, which stands for Code of Federal Regulations, Section 312. So I'm sure you'll all remember that, later on today, right? So there'll be a homework test, and right there, I'm looking at you in the back, They're required for all clinical investigations of biologic and pharmaceuticals, and they contain 5 modules. So when you're working with, whether it's an academic investigator, those usually end up with ISTs, or investigator-sponsored trials, or a company sponsor under the INDs. You still have to submit these as 5 modules. So they've got general information, all preclinical studies, chemistry, manufacturing, and controls, remember CMC, study reports, all of the preclinical study reports, and then your clinical protocol and informed consent. And it takes months often to put this together, even with an experienced team, because you have to integrate safety data, efficacy data, all of your manufacturing. Again, remember that's 21 CFR 312. You have to follow all of what they're looking for. When you submit this in the United States to the FDA, you've got 30 days, to return comments, for the FDA to return comments. If they return no comments, generally speaking, you're allowed to start your clinical trial. What often happens is that the FDA comes back on day 29 with about 16 pages of questions. And you've got 24 hours to go in and answer those questions with the 3 reviewers that you're trying to get a hold of, so that you're not either put on clinical hold or which starts a different clock about trying to get these. And these, this is a huge milestone for small companies. Getting an IND allowed is a big, big challenge, again, but that's a big value driver. And here's how you see how things kind of interact, with the clinical sites. And remember with some of the abbreviations, IRB and IBC, right? So Institutional Biological Safety Committee and Institutional Review Board. Those work within the clinical sites. The sponsor, usually a company or an institution, works with the sites. You've got to go through the FDA, which is the bracket around it. You've got to have your drug manufacturer and often your contract research organization. That's going to be the group that you go to help run your clinical trial. And again, we talked a little bit about our CMC. So again, when we talk about drug manufacturing, this goes into drug substance or drug product. So drug substance is you make everything in your microwave, and then you put it into a specific purified container. Once it's in that container, it becomes your drug product. So again, trying to, I see you, yeah. So the clinical site, there's a lot of participants that go into the clinical site. There's the sponsor, there's the principal investigator, there's the research coordinator. Every one of these plays a key part, in your clinical trial. And again, of course, you're trying to help accelerate your enrollments going through. So some considerations in clinical trial design. These place additional constraints on your clinical trial. So you're trying to determine what are the goals of the trial, how do you describe the drug, and what does the trial design look like? Number of patients, for example. Patient selection, who's going to potentially benefit, where is the safest population, where are things you need to think about for risk. There could be things like braces. One of the exclusion criteria in our clinical trials is braces because they all get MRIs, and if you have braces with an MRI, your teeth will get ripped out. Not a good, that's generally considered an adverse event. So the dosing schedule, are you gonna have a single dose or multiple doses? Inclusion criteria, that helps define the population, and exclusion criteria. So for example, for a lot of the AAV gene therapies, one of the tests that you have to do before a patient can be enrolled is test to see if they have antibodies against the virus. Okay, so, and we talk a lot about viral gene therapy and what's the percentage of patients that may have already existing antibodies against the AAV. That's a constraint of your clinical trial. Safety endpoints, again, going in to describe, the number of adverse events or the type of adverse events that can cause your data safety monitoring board to put a hold on your trial. So I know I'm throwing a lot of things out there, a lot of terminology out there, but this to also help set up, Ellen for some of her talk as well. So we'll skip through, some of that. And again, this is an example of an inclusion criteria. I think Ellen's going to talk a little bit more about that. Same thing with an exclusion criteria. This is just a laundry list of things that if a patient meets one of these things, they're going to be excluded. And again, Ellen's going to talk a bit more about that. So, and then you get into all of your outcome measures and assessments. And with that, I think I'll turn it over to Ellen.
Okay.
All right.
Thank you. So I am going to talk a little bit about some of the things that Tim mentioned in a little bit more detail. So first of all, I think a lot of you that may be in the advocacy arena and getting started in clinical trials are kind of wondering, why is it so difficult? Why is it so darn hard? Why does it take so long? So those are the things I'm going to touch on relative to the clinical trials in particular. So we all hear about the FDA, that the FDA requires this or that or the other thing, and they sound like the bad guy, but really the FDA is really just trying to protect the patients. And Tim was giving some information about the history of clinical trials, and we actually know a woman, Eva Moses-Core, who she's come to speak with our employees, who was a clinical trial subject during the Nazi occupation, and she was a twin. And so they were, I think she and her sister were about 5 or 6 years old, and they were subjects in this many clinical trials over several years. And she's a real advocate for making sure we protect the rights of patients. And that's really what the FDA is trying to do. They want to make sure that the drug is safe and that the treatment works. Okay, so how do I advance the is it up here?
This button right here. That one.
Okay, all right. Okay, so why do we need so many studies? So as Tim kind of mentioned, there are multiple types of clinical trials. You typically would do a phase 1 clinical trial where the objective is to establish a maximum and safe dose. And these studies are usually in the neighborhood of 20 to 40 patients, and sometimes they're in healthy, normal volunteers. And then if that phase is successful, then you would move on to phase 2, where the main objectives usually is to establish what the effective dose range is and to start to get a better understanding of treatment effects. And then phase 3 studies are your larger studies to prove that the drug is safe and effective, and can be anywhere from 50 to a few thousand patients. Now this is definitely can be different in rare diseases, especially ones that are more rare. Not the case with some of the more common rare diseases like cystic fibrosis. These types of trials are still required for that. Tim mentioned study endpoints. So it's really important to make sure that each study have a defined study endpoint. It's usually something that you measure, and you need to specify it in advance in your protocol, and you need to prove it. So examples of the primary endpoint might be something like the decrease in the cholesterol level measured by a blood test at, let's say, 6 months after starting treatment, something like that. So, and a little bit about study designs. There are lots of different study designs, and the FDA will generally work with you on determining what the best design might be for a rare disease trial. And I'm just going to mention a couple of the more common ones. There's a dose escalation study design where you may start patients out at a low dose, observe them, make sure it's safe, then move on to a higher dose with more patients, and then the next higher dose, and so on. Parallel studies where you would be studying active versus control, and they're receiving the same treatment at the same times, basically. And then crossover studies that are more complex in design, but can yield a lot more data with fewer patients. And in a crossover, certain patients would receive the study treatment first, and then wash out, and then cross over to the control treatment. And you tend to, from a statistical point of view, you are able to achieve more power with a crossover design. So, you know, one of the frustrations we see a lot, especially on the rare disease side, is why does the study need to have a control? Why does it have to have a placebo control? Why can't everybody get the study treatment? And, you know, big part of this is the placebo effect. So if people believe that they're getting the study treatment, they tend to think that they're improving, or they might tend to think that every little side effect they have is related to the study drug. And so it's important to have a control group so that you can kind of compare what sorts of things are being reported in one versus the other. A lot of times, especially in rare disease trials, you can use historical controls, but they may have different characteristics than the patients that are actually enrolled in the study trial. So that is something that I think is worth looking at, especially in rare disease trials. If you are doing a natural history study, it can be helpful to try and utilize that data as a historical control. And in that case, you might be able to have a smaller proportion of control patients in the actual trial. So, but it is best to have a control group and qualify patients in the same manner. And as I was saying, it is very frustrating sometimes that you have to have a placebo group or a control group in a clinical trial. In fact, we're working on a study where it's an ultra-rare disease, and there are actually only 10 patients in the study, and there's study sites in the US, and we identified a patient in Canada, and we have no study sites in Canada right now, and a consideration was given to fly the patient to one of the sites in the US. Health Canada would pay for that patient to come to the US if they were assured that the patient would get study treatment, but FDA was requiring that there be a placebo-controlled study, so Health Canada wouldn't pay for that patient. So in the end, we're setting up a site in Canada just to treat this one patient who may still get placebo, but hopefully that won't be the case. Tim kind of talked a bit about controlled clinical trials, so I won't go into that a whole lot. And a little bit about blinding. So this is one of the big constraints of clinical trials as well. So as you probably all know, most clinical trials are blinded, and neither the patient or the physician or the family knows which treatment a patient is receiving. And the reason that we need to do this is to remove bias, because if the study's not blinded, then again, if there's a side effect, the automatic assumption would be, well, it's related to the study drug. If the patient seems to be improving, automatically you would assume that it's related to the study drug. So by blinding, you help to remove some of that bias. And then why are treatments randomized? And that kind of goes along with the blinding. When you randomize, you reduce the possibility of bias and being able to guess which patient is getting which treatment. And that also helps to kind of make sure that you're not over-reporting side effects for one treatment versus another. And if the study was not randomized, the physician may maybe even unconsciously decide that, okay, I know that the next treatment is going to be active. I think I'm going to make sure that this particular patient who is maybe sicker than the others would get that. So we want to make sure that the treatment order is randomized so that that type of thing can't happen. And why do we have to have so many patients? And again, in rare trials, sometimes it can be a smaller patient population because the patients just aren't available. But the reason is that when you're doing a clinical trial, you're normally looking at just a sampling of the available patients or a sampling of the actual patients. And the smaller the sample, the greater the likelihood that you won't get a good representation of the population as a whole. You may get a group of patients who are sicker and less likely to respond. And that's really comes into play with types of studies where there may be an event-driven endpoint, such as a MI study, for example, where when there's an event and it's only maybe happens in 1 out of 20 people, and you happen to get slightly sicker patients in one treatment group than the other, the study could fail just because you've got sicker patients in the one trial, in the one arm. So in general, the larger the sample, the more representative it's going to be of the population as a whole. So, and just a little bit about error in clinical trials. And this is a big part of designing the study and choosing how many patients you need to enroll. So there's basically 2 types of errors that can occur in clinical trials. Type 1 is when there's really no effect, but the statistical test actually shows that it is significant, and you're assuming then that the treatment works when it really doesn't. And then the type 2 error is when there really is a treatment effect, but you don't have the right population, or you're not studying the right thing, or your sample is not large enough. And this is usually a sampling error issue. And this is probably what happens most often in clinical trials. If you've heard about p-values, you generally need a p-value of less than 0.05, which means that you have less than a 5% chance that anything that you see is due to error. And a lot of times, studies will miss. You'll end up with a p-value of 0.1, and then the FDA won't approve it. But they want to make sure that the trial, it really is effective and that your drug works. So in general, the more patients you can have in the study, the better. But you also have to take into account how many patients are available. Because statistical significance will be greater when you have more patients. And if you have more patients, the impact of unusual situations, exceptionally sick patients that have no chance of responding, they're minimized with a larger population. So, and as I mentioned, the p-value helps define the likelihood that the study results are not due to chance alone. So as we've discussed, there are a lot of exceptions for rare disease trials. We're working on a project right now in an ultra-rare disease where there was one patient that has been treated in a compassionate use. And the FDA actually gave the study sponsor permission to go ahead with just one clinical trial. So no phase 1, no phase 2. They went right into phase 3, and it is enrolling 10 patients only. So 5 in the active, 5 in the control. I don't think that happens very often, but because this disease is so rare and also so very serious, the FDA has agreed to this. They do want long-term safety data, and with a sample that small, there's a very high likelihood of failure, because if there's one patient in the placebo group that does well, or one patient in the active treatment that doesn't do well, the study won't show statistical significance. But the FDA will work with the sponsor on what to do next if that happens and the product still seems to show promise. So in addition, I mentioned some of the common study designs. There are definitely alternative study designs for rare diseases, such as randomized withdrawal, et cetera, different variations of crossovers that really can maximize the amount of data that you get with a small population. And as I mentioned, sometimes you don't have to do the full gamut of clinical trials. So, and oftentimes fewer patients. And the greater the treatment effect you expect to see, the fewer number of patients are typically required. So, and then sometimes the FDA will provide conditional approval on some small studies, and then they want, or they will require that then you provide some post-approval data. So continue to do some studies once the drug is approved. So wanted to see if anybody has any questions about anything.
I'm sure there's a ton of questions, right?
Thank you very much. So we have mics in the room, and you can also feed your questions in via the app. Does anybody in the room have any questions? I'm going to check here. You guys were so thorough that nobody has any questions. Oh, we've got a few. All right. Come on down. I feel like I'm in Price is Right.
Hi, Steve Grossman, ADCY5.org. One of the things that we're looking at is a natural history study with a lot of international participants. I think you kind of talked about that a little bit, but can you give us some thoughts on that?
So you talked about natural history.
Sure. So can you be just a little more specific when you say you're doing? So it's just natural history study?
It'd be a natural history study that hopefully lead to some more advanced studies. But I guess what do we need to take into consideration at this early stage as a small ultra rare disease with probably 40% of our population international? Sure.
So you're going to want to really start to compare a lot of the demographics first. Your ages. Some of their, real simple stuff like weight when they presented, what's their genetic diagnosis? That's going to be probably one of the bigger ones about, looking at when you're trying to do therapies and establish therapies, there's going to be a lot of different modifiers. So sometimes you're going to have fast progressors, sometimes you'll have slow progressors or an attenuated phenotype. Usually sometimes drugs, target different versions of those diseases or those forms of the disease. So making sure that you've got, some of the genetics behind why or what is the underlying cause. I probably suggest you want to work with the regulators in any one of those particular countries that says, here's the disease that we're looking at, here's what we know of the natural history study. Can you work with us on the clinical trial design? So you go through the natural history studies to try and identify outcome measures that are going to be able to show some level of, if you can intervene, that there may be some benefit in a reasonable time frame.
Okay, great, thanks.
Yeah, one other thing I just wanted to add too is standard of care. So kind of making sure that you understand the standard of care in different countries, because that can be very variable when you're looking internationally.
Yes, go ahead.
Thank you. Sean Gill with Pharma Clinical. So you did a great job of walking through, I think, the drug development plan, but I specifically want to talk about the protocol development. And with rare disease and patients being not centralized, perhaps in one location, what solutions do you incorporate for accessibility to get to these patients during the protocol development stage?
So are you asking specifically about how to get input from the patients during the development stage?
Or more specifically, how you lessen the patient burden during protocol development?
So during this study conduct?
Yes.
Yeah. So, I mean, some of the things that we do is, since a lot of rare disease patients may not be right around the study site, we provide a lot of assistance with providing home health care, so they don't have to come into the office so often. We also provide patient travel assistance, and then sometimes set up satellite sites that might be closer to where the patients are located, so they don't have to travel so far.
Yeah, Michelle, can you raise your hand for just a second? So right in the front up here. So Michelle is our Vice President of Patient Advocacy, and she's done a really great job of doing much of that, setting up for our clinical trials, patients coming into clinical sites. So she's done things like set up an apartment, because when they come in, they have to stay for the first from screening through like a day 30. That's about 45 days sometimes. So, trying to work with the hospital, the coordinators, and then an apartment complex that understands, that you can work with from a rare disease standpoint. Do they need wheelchair access, closeness to the hospital, transport to the hospital, things like that. And then, there's travel agencies that, focus on clinical trials. So if you want, Michelle can, certainly, give you some more advice on, and some context about how to go about that. Sorry, Michelle, throwing you out there.
Now she's got more work to do.
That's right.
Thank you both. It was a great presentation. Question I have for you is thinking about natural history studies and in clinical trial design. How many participants in a natural history study do you theorize may be required to make that natural history study powerful enough to serve as a control? Or is this a function of multiple things such as heterogeneity of the disease, incidence, prevalence? Are there many factors involved? Or is there sort of a magic number?
Well, that's an interesting question. So, beauty's in the eye of the beholder on that one, Tracy. It's going to, it depends on the disease. If you're looking at something that's very narrow, especially if it's a fast progressor, I think the one that at least immediately comes to mind is SMA. That's a very fast disease with a 90% infant mortality by the age of 3. So you don't have a lot of time to study, and survival becomes a very easy endpoint, frankly, in a natural history study. But ones where it's a little bit more difficult to measure, and usually a neurological or a neurocognitive score, you're going to need to plan that study to go out for at least a year, and you're going to have to measure those endpoints through there. So, and that's where you start to get into a little bit more of the subjective nature of saying, well, how much of a point of change on a slope do you need to see, and how many patients do you need to actually be able to show that to show that if you intervene, that you're going to be able to have a treatment effect. Compared to things like enzyme activity or a biomarker, where oftentimes it's either so much of it that if you can attenuate it at all, that's a treatment effect, so the sample size is small, versus if you're adding something back in and showing, oh, we're seeing 1,000-fold more expression, or more enzyme or protein in the blood, that's another treatment benefit, so you don't need as much to see that. So it's really going to depend on that, but it also goes back to working with the FDA or the EMA to say, we're thinking about doing a 20-patient natural history study. It's going to go on for 1 year, and here's the things we're going to look at. What do you think? And get their input to ask about.
The other thing regarding natural history studies, if you want to use it as a historical control, it will be important to conduct the study in as controlled a way that you can, where you're really specifying what tests patients should have and when, and then try and sort of match the patient population to the one that you'll be studying when you get to the clinical trial stage, so that they're actually comparable populations.
Okay, thank you.
Hi, Scott Schliebner, PRA Health Sciences. Thank you guys for the presentation. Quick question for you on kind of experiences you've had, positive examples around including patients up front, kind of a seat at the drug development table with, in terms of including patient input for protocol design, schedule of assessments, eligibility. What do patients want to see, maybe even endpoints that are meaningful to patients?
Yeah, sure. Absolutely. We have pretty much a standing mandate at Abeona to bring families with us to any regulatory meeting if possible. So that the regulators in particular can understand some of the face of the disease, and we have found that to be incredibly helpful to the FDA. It also gives the patients a voice, to the agencies often, but then you can also start talking with them about patient-reported outcomes, which many of these trials rely on to show some level of benefit. So, getting them involved early about what might be meaningful is really important because, as I think many of the families will tell you, their risk tolerance is much higher than many of the regulators, would. So communicating that very early about what may be a benefit, a really easy one is sleep. Many of the families, their children don't sleep very well. And so, the ability to have a study drug, that has as a benefit the ability to sleep for 6 uninterrupted hours, sometimes it's a little hard to measure, but that's a potentially very beneficial outcome. So yeah, bringing them along, with you is in your protocol design is often a key component.
All right, well, if there aren't any more questions, please
Actually,
join.
Sorry.
Yeah.
I just had one more question that I'd like to ask. Earlier this morning, there was some discussion about some of the man-made problems, or human-made problems, I guess we should say, related around drug development. And I was wondering if you could both comment on some of the things in clinical trials that are constraints that perhaps we could think about trying to shift so that they aren't so constraining, particularly for rare disease.
Well, one of the big things that we run into is getting sites initiated, and the contracting process with particularly academic institutions is very lengthy, and sometimes the review of the informed consent can take some time, and agreeing on a budget. And I don't know how to fix that, but I would love it if there could be something. I do understand that there is now a consortium of some of the academic sites that are coming up with a template contract. And once more of them implement that, it could make that whole process simpler. That's certainly the one that comes to mind quickest for me. Do you have any other thoughts on man-made?
Sometimes one of them is really finding clinical investigators that have expertise in a particular disease. One of the constraints of rare disease clinical trials is there's not a lot of PIs necessarily that have expertise in viral gene therapy and a lysosomal storage disease. You can count them on really one hand. There's a difference between the treatment in a clinical trial and then trying to do some of the translating the preclinical research. So I'd say that's one challenge. I mean, you're kind of asking about the human element, right? About just what can we, as on whatever role you fill, try to find ways to streamline clinical development. I mean, the FDA is doing a really good job right now. They're human too, but the number of orphan drug applications, I mean, you read this in the news, right? The number of orphan drug applications that have gone in or orphan products, has skyrocketed. So, they've had to move out their timelines and put in new processes to try and help with that. Part of it is just plan ahead. You got to plan ahead for a lot of that stuff.
The thing I think that really slows down clinical trials the most, though, is patient recruitment. And particularly with rare diseases, is finding the patients for the clinical trial. So that's something I think that patient advocacy groups can really help a lot with their registries. And being able to refer these patients to the clinical trials when they happen.
Great. Well, thank you both
There's
very much.
one more question coming up.
Jeff Levinson from the APBDRF. We're a patient advocacy organization. When you said eligibility, it sort of a key question. I don't know if you have any experience, say when you're dealing with a rare disease in a small patient pool, and you have more than one drug candidate. Could you talk a little bit about the complications in terms of eligibility when you're going from one to the other, washout periods or something like that?
Are you considering doing both drug candidates within the same trial?
So what's happening for us is that we're going to be running there will be a clinical trial outside the United States, and we're considering candidates in the United States. The one outside the United States will happen sooner. It'll be very difficult for the patient population in the United States to get there, so they'll obviously want to have a clinical trial here, probably with a different drug candidate. So will there be complications in terms of the eligibility if they start and go out of the country for the first clinical trial?
The same patients?
Yes.
Well, typically you'd want them to finish the first trial. If they live in the United States and now you open a site in the United States, but it's really part of the same trial, then they can transfer to a U.S. site.
We're actually thinking of a different drug candidate.
Oh, different drug
inside
candidate.
the United States.
No, then they would just need to, there'd need to be a certain amount of time in between the two studies for the patient.
And that would
30
be
days
sort of,
or 60 days or something.
Oh, is that right? Okay.
And usually there's an exclusion criteria that says that if you're in one trial or you've been within one trial, that you're ineligible to join another trial for depending on a time frame. So, for example, if you've received one gene therapy using a certain vector, you're basically going to be excluded from doing a very similar trial until you have a number of tests done. So or you're going to be trying to enroll patients into two very similar trials. So usually it's the clinician's job to go in and educate and say, you're going to have to make a choice about which trial you want to go into.
Thank you.
Great question. Thank you. Any others? We still have time. All right. Well,
Clinical
please join.
trials.
I'm sorry.
Anything else? Clinical trials.
It's late on Friday.
Yeah.
Well, please join me in thanking our speakers today. You'll now have 10 minutes to transfer to your next breakout session, but as a reminder, some of the rooms are no longer hosting sessions, so please just refer to your agenda. Thank you again and enjoy the rest of your time.